Hello bios, welcome to Decoding Biology 111. I know you're all excited to know why the thumbnail for this video is given little different. Yes, I know that it is always adenine which pairs with thymine, and it is guanine which always pairs with cytosine. That's what we see in DNA. But let's try to ask some interesting questions towards this and try to understand this complementary base pairing concept a little deeper. So in DNA, as we all know that it is adenine, which is a purine, pairs with a pyrimidine, that is thymine, and guanine, again which is a purine, pairs with the pyrimidine cytosine. We see this complementary base pairing in DNA. Now, to address the thumbnail question, let's try to understand first what is the meaning of complementary base pairing. If two bases could join and form hydrogen bonds between them, then we call that as complementary base pair. In DNA, we don't just talk about complementary base pairing, but we do talk about a specific complementary base pairing, meaning it's not that any purine will be pairing with any pyrimidine. Say for example, A can sometimes pair with T and sometimes with C. No, it's not like that. And again, G, sometimes it pairs with G and sometimes it pairs with C. It's not like that. A always pairs with G and G always pairs with C. This is called as specific complementary base pairing, which is seen in DNA. Now let's try to understand why we see this specific complementary base pairing, that is AT and GC. Why not A with C pairing with C and G pairing with T? Now, if you look at the structures of the nitrogenous bases which are given in the corners, the structure of A with all the side groups, with the side attached groups, are in such a way that if it combines with T, it could form two hydrogen bonds. And in the same way, the structure of guanine is in such a way that if it could combine with C, it could form three hydrogen bonds. But if A wants to pair with C, can A form a pair with C? Yes, it can form a pair with C, but the number of hydrogen bonds possible between A and C is only 1, not 2. Same way, G also can pair with T, and again the number of hydrogen bonds possible between G and T is 2 but not 3. So which is good? 2 hydrogen bonds between AT and 3 hydrogen bonds between GC is good, or 1 hydrogen bond between AC and 2 hydrogen bonds between GT is good. Obviously, the 2 hydrogen bonds between AT is good and 3 hydrogen bonds between GC is good compared to the other part we discussed, compared to AC pair and GT pair. That is why DNA, which is the smartest genetic material, has chosen the specific pairing, that is AT and GC, so that it could have many hydrogen bonds between the two strands. Hope you liked this explanation. For more such concepts to be clear, do subscribe my channel Decoding Biology 111, and I'll meet you in the next video with an interesting fact. Thank you for watching.